Hey everyone, welcome back for another deep dive. You know, we're all about staying ahead of the curve on the most important health topics. And today we're going somewhere pretty amazing.
Hmm.
We're talking about the potential for mRNA technology. You know, the stuff that brought us the COVID vaccines to actually revolutionize HIV treatment.
Yeah, maybe even lead to a cure.
A cure.
Ah,
I mean, that might sound like a giant leap, especially for those of you familiar with our focus here. You know, connecting you with trusted HIV testing resources like our massive network of over 4,500 testing labs all over the US.
right.
But the science is there. It's real. And we're diving deep into exactly how it works.
And what it can mean for all of us, especially those who want to stay informed quickly and thoroughly. The research is really exciting, offering a hopeful new perspective on the future of HIV care.
Okay, so HIV, we've all heard about it for decades, but let's rewind a bit. Can you give us the download on what HIV actually does in the body and why it's been such a tough opponent to beat?
Definitely. So HIV, which stands for Human Immunodeficiency Virus, is a virus that attacks the body's immune system. Specifically, it targets a type of white blood cell called a CD4 plus T cell. These CD4 cells, they're like the conductors of the immune system's orchestra. They organize the other immune cells to fight off infections.
Oh, they're essential.
Exactly. And when HIV comes in, it infects these crucial cells and over time destroys them.
Oh, wow.
And without enough CD4 T cells, the body is left wide open to infections. Things that wouldn't normally be a threat become very dangerous, and certain cancers can also take hold more easily.
That's scary stuff. So it's not just a quick attack. It's like a takeover from the inside. What makes it so difficult for our immune system and even current treatments to completely get rid of HIV?
Well, there are a few big reasons for that. First, HIV is sneaky. It can create what are called latency reservoirs. Think of these like hideouts where the virus can go dormant, hiding within certain immune cells, especially in memory T cells.
So they're invisible.
Essentially, yeah. While HIV is in this inactive state, it's undetectable to the immune system and most antiviral drugs. It's off the radar. Then you've got HIV's incredibly high mutation rate. Every time it replicates, it makes mistakes, little changes in its genetic code. This leads to a huge variety of HIV within a single person.
Like it's constantly changing its disguise.
Exactly, and that makes it super hard to design a single therapy that can target all these different forms.
So it's kind of like playing whack-a-mole with the virus.
Yeah, a really fast and complicated game of whack-a-mole. And then there's the biggest challenge of all. HIV actually integrates its own genetic material, its DNA, into the DNA of the host cells it infects.
So it becomes a part of us.
In a way, yeah. It's a very unwelcome guest. This integration makes it incredibly difficult to get rid of the virus completely without harming the healthy cells, too.
Okay, that paints a pretty clear picture of just how tricky HIV is. We do have antiretroviral therapy, RT, which has been life-changing for so many people. It allows them to live long, healthy lives. But it's not a cure, right? Can you explain how ART works and why it falls short of actually eradicating the virus?
Sure. So ART is a huge achievement in managing HIV. It works by suppressing the virus, bringing down the amount of HIV in the blood, what we call the viral load, to very low levels, sometimes even undetectable levels.
Okay.
This lets people with HIV live long, healthy lives. And it also prevents transmission to others, a big win. But here's the catch. ART doesn't completely eliminate HIV from the body.
Especially from those reservoirs, those hideouts you mentioned earlier.
Exactly. So even with ART, HIV can be lurking in those dormant cells. And that means people usually have to take medication every day, potentially for life, to keep the virus under control.
To keep it from coming back.
Exactly. Plus, RT can have some side effects for certain people, and access to consistent treatment remains a major issue in many parts of the world. Even in places with good healthcare systems like the US, ensuring equitable access to testing, like our network of over 4,500 labs, is a constant challenge.
So ARC is doing a great job managing HIV, but a true cure is still what we're aiming for. So let's talk about mRNA. We all know it now from the COVID vaccines. But for someone who might just know the basics, can you break down what mRNA technology actually is and why it holds so much promise in medicine?
Sure. Think of messenger RNA, or mRNA, as a set of instructions for our cells. Our DNA, which is our genetic blueprint, contains the code for all the proteins our bodies need to function. But when a particular protein is needed, the cell makes a copy of the relevant DNA instructions in the form of mRNA.
So it's like a messenger.
Exactly. This mRNA then travels out of the cell's nucleus to the protein-making machinery called ribosomes. And the ribosomes read those mRNA instructions and assemble the specific protein. So mRNA technology uses this natural process, but with the twist. Scientists can create synthetic mRNA that carries instructions for the cell to produce any protein they want, like, say, a harmless piece of a virus.
So we can tell our cells what to make.
Yeah. And in the case of vaccines, our immune system sees this foreign protein and learns to fight it off. So the mRNA is like a temporary code that trains our immune system.
That's amazing. It's like giving our cells a recipe to cook up a defense against disease. And the COVID vaccine showed us just how fast mRNA technology can work. Does this speed and adaptability give it an edge against HIV, which, as we discussed, is really good at mutating and evading the immune system?
Definitely. The mRNA COVID vaccines were developed and rolled out at an unprecedented pace, and that really highlighted the speed and flexibility of this technology. Unlike traditional vaccines, which can take a long time to develop, mRNA vaccines can be designed and produced pretty quickly once you know the genetic sequence of your target.
So we can target new variants more easily.
Precisely. And with HIV, which is constantly mutating, that's huge. As new concerning strains pop up, scientists could potentially tweak the mRNA code in a vaccine pretty quickly, maybe in a few weeks. That gives us a real fighting chance to keep up with HIV's evolution, something previous vaccine attempts have struggled with.
Okay, so now let's get to the exciting part. Applying all this to HIV. Our source material talks about some groundbreaking research happening with mRNA HIV vaccines. What's going on there?
One of the most promising areas is the development of mRNA vaccines that can trigger our bodies to produce something called broadly neutralizing antibodies, or BNAbs for short. These are a special type of antibody that can recognize and neutralize a wide range of HIV strains, not just one specific version.
So instead of teaching our immune system to fight just one type of HIV, we're teaching it to recognize and fight many different versions at once.
That's the goal, and that's been a huge focus in HIV vaccine research for years. And Moderna, the company that made one of the most effective COVID vaccines, is actually running trials for an mRNA HIV vaccine called mRNA 1644.
So they're in the game now.
Big time. They started these trials in early 2022, and the goal is to deliver the instructions for our cells to produce proteins that trigger the creation of these broadly neutralizing antibodies. And it's not just Moderna. The National Institutes of Health, or NIH, is also investing heavily in this research.
So a lot of brain power going into this.
Oh, yeah. The NIH is teaming up with a bunch of different pharmaceutical companies to advance mRNA vaccine development for HIV, and the early results are really promising, especially in animal studies with primates. They're even starting initial trials with humans.
Wow. So how do these vaccines actually work against HIV?
It's pretty cool. The vaccines deliver mRNA that carries the code for making an HIV-like protein.
A decoy almost.
In a sense, yeah. And this tricks the immune system into thinking it's encountering the real HIV virus.
So it mounts a defense.
Exactly. This triggers an immune response, hopefully leading to the production of antibodies that can then block the actual HIV virus from infecting cells.
Okay, so we've talked a lot about vaccines, which are about preventing HIV infection. But the bigger question, the one I think everyone's wondering about is, could mRNA technology actually cure HIV in people who are already living with the virus? Is that even possible?
It's a big question. And it's important to be clear about the difference between a vaccine, which prevents or controls infection, and a cure, which means getting rid of HIV from the body completely.
Eradicating it.
Exactly. But the research is pointing towards an incredibly exciting possibility: combining mRNA technology with gene editing tools, specifically CRISPR.
Oh, CRISPR. That's the one that acts like molecular scissors, right?
Yeah.
It can cut and edit DNA with incredible precision. How would that work for HIV? And where does mRNA come into play?
You got it. CRISPR lets scientists target and cut specific DNA sequences. And the idea with HIV is to use CRISPR to cut out the HIV DNA that's integrated itself into our cells to remove it from our genetic code.
So like a delete key for HIV.
Yeah. And in 2023, there was a breakthrough. Researchers actually showed that they could use CRISPR to cut HIV DNA out of infected cells in the lab.
Wow.
That's huge, but there's a big challenge. How do we deliver this CRISPR system safely and effectively to all the infected cells in the body, including those hidden away in those latency reservoirs?
It's a delivery problem.
Exactly, and that's where mRNA comes in. Scientists are exploring using mRNA as a delivery system for CRISPR.
Like a tiny courier.
Yeah. The mRNA would carry the instructions for the cell to make the CRISPR components, and then those components would edit the cell's DNA and remove the virus from within.
So we're using mRNA to tell our cells to cure themselves.
That's the idea.
But even if we could edit the virus out of most cells, we still have those tricky latency reservoirs to deal with.
Absolutely. Those reservoirs are the biggest obstacle to a complete cure. Even if gene editing worked perfectly in most cells, the virus hiding in the reservoirs could reactivate and start the infection all over again. So a true cure would have to target those reservoirs too, maybe by getting rid of the latently infected cells or finding a way to keep the virus from ever waking up again. It's a complex problem.
A multi-pronged approach.
For sure. But the science is moving fast.
No.
And there's a lot of optimism that these new approaches, combining mRNA and gene editing, could accelerate the search for an HIV cure, maybe even faster than we thought possible.
From what we've read, some experts are even predicting a functional cure within 10 to 15 years.
That's right. And while that's still a prediction and research timelines can change, it's an incredibly hopeful outlook. Just a few years ago, this wouldn't have even been on the table.
That's really encouraging. But taking these breakthroughs from the lab to the real world, making them accessible to everyone globally, that's a whole other challenge, right? Especially for our listeners who are navigating the world of HIV testing and treatment.
You're absolutely right. There are a lot of hurdles. Cost is a big one. These new technologies are expensive to develop and produce, which could make them out of reach for people in low-income regions where HIV often hits the hardest and where even basic testing can be scarce.
That's a huge concern.
It is. Then there's the challenge of accessibility, making sure these treatments reach everyone who needs them, regardless of where they live or their economic situation. And we can't forget about the stigma that still surrounds HIV. We need to keep fighting that stigma and the misinformation that goes with it, because it can prevent people from getting tested, sticking to treatment, and embracing new prevention and cure strategies.
So it's not just about the science, it's about social and economic factors too.
Exactly. We need to work on all those fronts to make sure these breakthroughs actually benefit everyone.
Okay, so for our listeners who are interested in staying informed and proactive about their health, what steps would you recommend?
Staying informed is key. Find trustworthy sources of information about HIV prevention and treatment so you can make informed decisions about your health.
Stay curious, stay engaged.
And don't underestimate the power of regular HIV testing. Early detection is so important. It allows for early treatment, which dramatically reduces the risk of complications and improves overall health. Plus, it helps prevent the spread of the virus. A lot of people don't show symptoms in the early stages of HIV, so testing is the only way to know for sure.
Absolutely. And for our listeners in the U.S. who are looking for fast, confidential, and convenient HIV testing options, we always recommend checking out HIVRNatetestguide.com.
It's a fantastic resource. They have this incredible network of over 4,500 testing labs all across the country, making it easy to get tested discreetly and comfortably right in your own community.
Excuses.
Knowing your status is the first and most important step towards protecting your health and the health of others.
Couldn't agree more. Now, beyond getting tested, are there other ways people can get involved in the fight against HIV, contribute to research, or help move us closer to a cure?
Participating in clinical trials is a huge help. These trials are always looking for volunteers to test the safety and effectiveness of new treatments and prevention methods. It's a way to be a part of the solution and potentially get access to cutting-edge treatments that aren't available yet.
So that's another great way to stay proactive.
Exactly. And you can usually find information about ongoing clinical trials through research institutions, advocacy organizations, and your healthcare provider.
All right, so wrapping it all up, it seems like mRNA technology is bringing a lot of hope to the fight against HIV. More effective prevention, maybe even a cure in the future. It's pretty exciting stuff.
It really is. mRNA has the potential to change how we approach HIV entirely. There are definitely still challenges ahead, but the progress we're seeing with mRNA vaccines and gene editing is really remarkable. It's opening up a real pathway towards preventing new infections and achieving a lasting cure.
And for our listeners, as we imagine this future, a future where HIV is no longer a threat, we want to leave you with a thought. If mRNA can do all this for a virus as complex as HIV, what other possibilities are out there? What other persistent viral infections could we tackle with this technology? Hepatitis B, herpes. It's a powerful question to consider as we continue to explore the potential of mRNA. The journey towards ending HIV isn't over, but the progress we're making is huge. It gives us real hope for a healthier future.
I couldn't agree more.
Thanks for joining us on this deep dive. Until next time, stay informed, stay curious, and stay well.